Section 23 of Anatomy of the Human Body, Part 5. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 5, by Henry Gray. The Liver, Part 1. Hepar. The liver, the largest gland in the body, has both external and internal secretions, which are formed in the hepatic cells. Its external secretion, the bile, is collected after passing through the bile capillaries by the bile ducts, which join like the twigs and branches of a tree to form two large ducts, which unite to form the hepatic duct. The bile is either carried to the gallbladder by the cystic duct, or poured directly into the duodenum by the common bile duct where it aids in digestion. The internal secretions are concerned with the metabolism of both nitrogenous and carbohydrate materials absorbed from the intestine and carried to the liver by the portal vein. The carbohydrates are stored in the hepatic cells in the form of glycogen, which is secreted in the form of sugar directly into the bloodstream. Some of the cells lining the blood capillaries of the liver are concerned in the destruction of red blood corpuscles. It is situated in the upper and right parts of the abdominal cavity, occupying almost the whole of the right hypochondrium, the greater part of the epigastrium, and not uncommonly extending into the left hypochondrium as far as the mammillary line. In the male it weighs from 1.4 to 1.6 kilograms, in the female from 1.2 to 1.4 kilograms. It is relatively much larger in the fetus than in the adult, constituting in the former about 1/18th and in the latter about 1/36th of the entire body weight. Its greatest transverse measurement is from 20 to 22.5 centimeters. Vertically, near its lateral or right surface, it measures about 15 to 17.5 centimeters, while its greatest antero-posterior diameter is on a level with the upper end of the right kidney and is from 10 to 12.5 centimeters. Opposite the vertebral column, its measurement from before backward is reduced to about 7.5 centimeters. Its consistence is that of a soft solid. It is friable, easily lacerated, and highly vascular. Its color is a dark reddish brown, and its specific gravity is 1.05. To obtain a correct idea of its shape, it must be hardened in situ, and it will then be seen to present the appearance of a wedge, the base of which is directed to the right, and the thin edge toward the left. Symington describes its shape as that of a right-angled triangular prism with the right angle rounded off. Surfaces. The liver possesses three surfaces, namely superior, inferior, and posterior. A sharp, well-defined margin divides the inferior from the superior in front. The other margins are rounded. The superior surface is attached to the diaphragm and anterior abdominal wall by a triangular falciform fold of peritoneum, the falciform ligament, in the free margin of which is a rounded cord, the ligamentum teres, obliterated umbilical vein. The line of attachment of the falciform ligament divides the liver into two parts, termed the right and left lobes, the right being much the larger. The inferior and posterior surfaces are divided into four lobes by 5 fossae, which are arranged in the form of the letter H. The left limb of the H marks on these surfaces the division of the liver into right and left lobes. It is known as the left sagittal fossa, and consists of two parts, namely, the fossa for the umbilical vein in front and the fossa for the ductus venosus behind. The right limb of the H is formed in front by the fossa for the gallbladder and behind by the fossa for the inferior vena cava. These two fossae are separated from one another by a band of liver substance termed the caudate process. The bar connecting the two limbs of the H is the porta, transverse fissure. In front of it is the quadrate lobe, behind it the caudate lobe. The superior surface, facies superior, comprises a part of both lobes, and as a whole is convex, and fits under the vault of the diaphragm, which in front separates it on the right from the 6th to the 10th ribs and their cartilages, and on the left from the 7th and 8th costal cartilages. Its middle part lies behind the xiphoid process, and in the angle between the diverging rib cartilage of opposite sides is in contact with the abdominal wall. Behind this the diaphragm separates the liver from the lower part of the lungs and pleurae, the heart and pericardium, and the right costal arches from the 7th to the 11th inclusive. It is completely covered by peritoneum, except along the line of attachment of the falciform ligament. The inferior surface, facies inferior, visceral surface, is uneven, concave, directed downward, backward, and to the left, and is in relation with the stomach and duodenum, the right colic flexure, and the right kidney and suprarenal gland. The surface is almost completely invested by peritoneum. The only parts devoid of this covering are where the gallbladder is attached to the liver, and at the porta hepatis, where the two layers of the lesser omentum are separated from each other by the blood vessels and ducts of the liver. The inferior surface of the left lobe presents behind and to the left the gastric impression, molded over the antero-superior surface of the stomach, and to the right of this a rounded eminence, the tuber omentale, which fits into the concavity of the lesser curvature of the stomach and lies in front of the anterior layer of the lesser omentum. The under surface of the right lobe is divided into two unequal portions by the fossa for the gallbladder. The portion to the left, the smaller of the two, is the quadrate lobe, and is in relation with the pyloric end of the stomach, the superior portion of the duodenum, and the transverse colon. The portion of the under surface of the right lobe to the right of the fossa for the gallbladder presents two impressions, one situated behind the other and separated by a ridge. The anterior of these two impressions, the colic impression, is shallow and is produced by the right colic flexure. The posterior, the renal impression, is deeper and is occupied by the upper part of the right kidney and the lower part of the right suprarenal gland. Medial to the renal impression is a third and slightly marked impression, lying between it and the neck of the gallbladder. This is caused by the descending portion of the duodenum and is known as the duodenal impression. Just in front of the inferior vena cava is a narrow strip of liver tissue, the caudate process, which connects the right inferior angle of the caudate lobe to the under surface of the right lobe. It forms the upper boundary of the epiploic foramen of the peritoneum. The posterior surface, facies posterior, is rounded and broad behind the right lobe, but narrow on the left. Over a large part of its extent it is not covered by peritoneum. This uncovered portion is about 7.5 centimeters broad at its widest part and is in direct contact with the diaphragm. It is marked off from the upper surface by the line of reflection of the upper layer of the coronary ligament, and from the under surface by the line of reflection of the lower layer of the coronary ligament. The central part of the posterior surface presents a deep concavity which is molded on the vertebral column and crura of the diaphragm. To the right of this, the inferior vena cava is lodged in its fossa between the uncovered area and the caudate lobe. Close to the right of this fossa and immediately above the renal impression is a small triangular depressed area, the suprarenal impression, the greater part of which is devoid of peritoneum. It lodges the right suprarenal gland. To the left of the inferior vena cava is the caudate lobe, which lies between the fossa for the vena cava and the fossa for the ductus venosus. Its lower end projects and forms part of the posterior border of the porta. On the right it is connected with the under surface of the right lobe of the liver by the caudate process, and on the left it presents an elevation, the papillary process. Its posterior surface rests upon the diaphragm, being separated from it merely by the upper part of the omental bursa. To the left of the fossa for the ductus venosus is a groove in which lies the antrum cardiacum of the esophagus. The anterior border, margo anterior, is thin and sharp, and marked opposite the attachment of the falciform ligament by a deep notch, the umbilical notch, and opposite the cartilage of the ninth rib by a second notch for the fundus of the gallbladder. In adult males, this border generally corresponds with the lower margin of the thorax in the right mammillary line, but in women and children it usually projects below the ribs. The left extremity of the liver is thin and flattened from above downward. Fossae. The left sagittal fossa, fossa sagittalis sinistra, longitudinal fissure, is a deep groove which extends from the notch on the anterior margin of the liver to the upper border of the posterior surface of the organ. It separates the right and left lobes. The porta joins it at right angles and divides it into two parts. The anterior part, or fossa for the umbilical vein, lodges the umbilical vein in the fetus, and its remains, the ligamentum teres, in the adult. It lies between the quadrate lobe and the left lobe of the liver, and is partially bridged over by a prolongation of the hepatic substance, the pons hepatis.